Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing TIFR Biology 2024 question paper. I have discussed questions from biology coming under the general section, from biology questions coming under chemistry section, as well as biology section. So these three sections I have discussed. If you are new to my channel, please do like, share and subscribe. I have also uploaded previous year TIFR biology question paper. You can go through the same in my channel. Coming to the first question. Which of the following disease is not caused by bacterial infection? So, anthrax is Bacillus anthracis, tetanus is caused by tetanus bacteria, syphilis is also caused by bacteria. Shingles is not caused by bacteria, it is caused by varicella zoster virus. Then next one. These are biology questions coming under general section. Earth's temperature is increasing as an effect of greenhouse effect. Which of the following gases does not participate in greenhouse effect? They have asked not participate in greenhouse effect. Methane is a greenhouse gas. Nitrogen is not a greenhouse gas. Carbon dioxide and ozone are also greenhouse gases. So answer here is nitrogen. Then next one. A bacterial population doubles every 2 hours. If the population size is n at this moment, what will be the size of the population after 2 days? So initial population is n, that is equal to, I have taken it as a. Then they have asked 2 days. 2 days, here the time is in 2 hours. So 2 days they have asked, so convert days into hours, that is 48 hours. So doubling time is 2 hours as per the given question. So 48 divided by 2 will be 24 generations will be present. The generation present I am representing it as small n. Then final population is given by the formula A into 2 raised to n. A is capital N into 2 raised to 24. So the answer here is n into 2 raised to 24. That is the second option. Then next one. Protein P function can be activated as well as inhibited by adding specific ligands. In an experimental setup, a student measured the expression level of protein P in arbitrary units and lysosomal pH of individual cells in different conditions. These values are plotted where the relative expression of protein P is plotted on the x-axis and lysosomal pH is plotted in y-axis. Values from each cell is represented by dots. The corresponding correlation coefficient values were also calculated. Based on this information, which statement is most correct? So if you see the graph, you can see the R value, that is correlation coefficient value, is approximately near to 1 in all the three graphs, showing that there is a relation between protein P, expression of protein, protein P and lysosomal pH. That is, protein P may function as a regulator of lysosomal pH. So we can see from the graph whether it is in the control condition where protein P is very much less expressed, or whether protein P is activated, or whether protein P is inhibited. It is functioning as a regulator of lysosomal pH. Now we will read the options. Protein P may function as a regulator of lysosomal pH irrespective of its expression. Yes, that is correct. Protein P, but you have to choose the most correct statement. That is what is asked in the question. So we have to see all the statements. Protein P may function as a regulator of lysosomal pH irrespective of its activation or inhibition. As per these two graphs I already explained now, it is also correct. Protein P may function as a regulator of lysosomal pH irrespective of its expression, activation or inhibition. That statement is more correct. Protein P is less likely to be involved. It is involved in the regulation of lysosomal pH. So D statement is not correct. Then coming to biology questions from the chemistry section. First question. The pKa of amino group of a Zwitter ion is 9.6. If 0.1 molar solution of the Zwitter ion at pH 9, what percentage of the amino group of the Zwitter ion is protonated? So here you have to use the Henderson-Hasselbalch equation. pH is equal to pKa plus log concentration of A by concentration of HA, where A is the deprotonated form and HA is the protonated form. Now substitute the values. 9 is the pH and 9.6 is the pKa. From there you have to find out what is the concentration of A by concentration of HA. So you have to take antilog of 0.6. So antilog of 0.6 is 0.2. Here I made a mistake of writing log over here. You need not have to write here once you have written antilog on this side. So concentration of A by concentration of HA. Concentration of A is equal to 0.2 concentration of HA. Concentration of A plus HA will be 1 molar. Substitute the value of concentration of A is equal to 0.2 HA over here. You will get it as HA is equal to 83%, or as per the options it is 80%. So the answer is option A, that is 80%. You have to remember the Henderson-Hasselbalch equation as well as concentration of A plus concentration of HA is equal to 1 molar. These 2 points, if you remember, you can solve this question easily. Then next one. During the process of oxidative phosphorylation, which molecule acts as terminal electron acceptor? That is a very easy question, and the answer, it is a direct question also, and the answer is oxygen. Oxygen is the terminal electron acceptor. Then next one. 2 strands of DNA double helix are linked to each other by. So 2 strands of the DNA helix. This is one strand and this is second strand. These are linked by hydrogen bonds between the bases. A to T has 2 hydrogen bonds. GC has 3 hydrogen bonds. So hydrogen bond between bases is the answer. Answer is option C. What does allosteric inhibition of an enzyme by a ligand mean? So what is allosteric inhibition? It is a type of non-competitive inhibition such that the inhibitor binds to a location on the enzyme other than its active site. So it doesn't bind to the active site but to another location which is not an active site of the enzyme. It will bind there, and that binding alters the overall shape of the enzyme such that it can no longer bind to the substrate. This is called as allosteric inhibition. So we will read the statements now. The binding of a ligand to the enzyme inhibits binding of cognate substrate at the same binding site. It is not the same binding site, it is at a different binding site. I told you already, allosteric inhibition is inhibition of the enzyme by allosteric inhibitor by binding to a location other than the active site. So it is not the same binding site, it should be different binding site. The binding of a ligand to the enzyme inhibits the binding of the cognate substrate at the different binding site. That is correct. Ligands binds with protein covalently. No, that is not the correct statement as per allosteric inhibition. All the binding sites are occupied by the ligand. That is also not correct. Then next one. In the case of fluorescence resonance energy transfer, that is FRET, F-R-E-T, first I will tell you what is the principle behind FRET. It is needed in protein-protein interaction studies. So one protein is bound to a fluorophore, that is cyanogen fluorescence protein CFP, and the protein B is bound to another fluorophore, that is yellow fluorescence protein. When these two proteins are not bound to each other, then fret will not be produced and the light will not be emitted. But once they are bound to each other, these proteins are bound to, these protein A and protein B are bound to each other, whatever excitation light produced by this CFP will be accepted by the YFP, and it will emit fluorescent light which could be captured by the detector. So fret is based on the fact that a donor dye in an excited state, this is the donor dye, CFP is the donor dye, in an excited state, that is upon excitation, can transfer a part of its energy through non-radiative dipole-dipole coupling. That is the mechanism by which energy is transferred from CFP to YFP when protein B and protein A interact with each other. Okay. Now what they have told is the energy donor has smaller excitation wavelength and energy acceptor has larger excitation wavelength. So this is the excitation wavelength of the donor and this is the excitation, the emitted wavelength of the acceptor. Now what they have told, the energy donor has smaller excitation wavelength. So CFP is having smaller excitation wavelength and energy acceptor, that is YFP, has larger excitation wavelength. That is correct in case of fret. So answer is 10A. Then next one. Protein X contains 192 amino acids and ubiquitinated at 2 residues. What is ubiquitination? It is when ubiquitinated molecule bind to a protein, it means that the protein is going to be get degraded. Okay, now that is not the question here. What could be the probable molecular weight of the protein? So once ubiquitin is attached to the protein, what will be its molecular weight? So here the protein X, it contains 192 amino acids. So molecular weight of one amino acid is 110 Daltons. So 192 amino acids will be approximately 21 kDa. Two ubiquitin molecules are added. They have given molecular weight of ubiquitin is 8 kDa. So one ubiquitin is 8 kDa. So 2 ubiquitin will be 16 kDa. So molecular weight of ubiquitinated protein will be 21 plus 16, that is 37 kDa. So the answer here is option C, that is 37 kDa. Then next one. A kinesin motor moves at 1.8 micrometer per second. If it is walking on a microtubule of 10 millimeter in length, how long will it take to cover the entire distance? This is just velocity is equal to distance traveled by time. That formula you have to use here, but see that you have to convert the units. 1.8 micrometer you have to convert into millimeter. That is 0.0018 millimeter per second. Now the distance traveled is 10 millimeter. So time you have to calculate. Time is equal to distance by velocity. If you substitute the values in the formula, you will get it as 5555 seconds. Now seconds you have to convert it into minutes and you will get it as 92.83 minutes. Or approximately 90 minutes as given in the answer. So the answer here is option B. Then next one. The molecular formula for amino acid glycine is C2H5O2N. What would be the molecular formula for a linear peptide made by linking 10 glycine molecules by condensation synthesis. So here they have already mentioned it is condensation synthesis that is taking place while peptide bond is formed. So you have C2H5O2N. Now 10 molecule of glycines are joined. So multiply by 10. 10 into C2H5O2N, you will get C20H50O20 and 10. Now peptide bond formation, 1 molecule of water is released. So here 10 molecules of glycine are joined. So 10 molecules of water will be released. So that will come up to C20H30, 50 minus 20 and O20. So O will become 10 and then 10. So that is somewhat same as C20, H32, O11 and 10. So the answer here is option C. Then coming to questions from biology section. First one. A dash hunt is a type of dog with short stature. Beagles are normal sized. The progeny of a dash hunt beagle mating are all short statured. Half of the puppies are produced with the beagle dachshund progeny when mated with beagle have short legs. The other half are normal size. What does this tell you about short stature? So first we will see what they have asked. So dachshund is a type of dog with short stature. So small s I will take it as short and the normal as capital S. Now, the progeny of a dachshund beagle mating are all short statured. So, if that is the case, if I take short stature as a recessive character and normal size as a dominant character, what I will get? I will get normal dogs. Normal sized dogs, not short stature. I made a mistake here. It is not short stature, it is normal stature. So it is not short stature, it is normal stature. You will get. Now what is the next thing they have told? Half of the puppies that are produced with beagle dachshund progeny, that is short statured progeny, when mated with beagle. So short statured progeny, that is here the normal stature whatever you get is mated with beagle, that is beagle I have given it as dominant character. So capital S I will get all normal progeny. But what does the question give you? They give you half of the puppies that are produced will have short legs and the other half has normal sized legs. So here beagle condition, that is normal sized condition, is not dominant, it is recessive, and short stature is dominant. Now if you take it in that way, normal is smallest crossed with short stature, that is capital S, you will get capital S small s, which is short stature. That is as per the question. The progeny of the dachshund beagle mating are all short statured. So you got short statured. Then you cross short with this F1 progeny with the normal. Then you will get half of them that are short and half of them that are normal. So short stature is a dominant trait. It is not a recessive trait. Answer is option D. Then next one. Which of the following options regarding an mRNA formed by bacterial transcription is correct? So this is the bacterial mRNA coding DNA. So this is the DNA. This is the promoter sequence. This is the DNA that is coding for this particular mRNA. This is the promoter sequence. This is the transcription start site. This is RNA coding region where you will find the start codon. This is the termination site. So here they have told the transcription start site occurs few bases before the start codon. So your start codon will be somewhere over here. This one, AUG. It will be somewhere over here, and a few bases upstream or before you have the transcription start site. That is correct. So the answer here is option A. Then the number of alleles possible for a gene is infinite number of alleles can be formed. Then how does an adaptation such as better running evolve with natural selection? Now if the adaptation for better running speed is beneficial to the survival and reproduction of the organism, then random mutations will naturally select it. Okay, that is how natural selection is working. So natural selection directionally produces better adaptation over time, gradually leading to running speed. No. Natural selection make individuals with favorable traits such as better running speed more attractive, ensuring they produce more offsprings. No. Natural selection involves random mutations. Yes. Some of which are beneficial adaptations. Heritable beneficial adaptation lead to offsprings with higher survival. That is more correct. So answer is option C. Then next one. Despite the large number of unique mutations described in LDRR gene that cause familial hypercholesteremia, certain mutations are found in high frequency in specific populations. Example, the 9.5 kb 3' deletion found in 35% of Finnish patients. This is an example of. Now here you have got a large population, large number of unique mutations for LDR gene that causes familial hypercholesteremia. But there are certain mutations, that is the 9.5 kb 3' deletion found in Finnish persons from this larger population. There is a very small number of individuals showing 9.5 kb 3' deletion from among this larger population. That is, this is the larger population, the shaded one, and this is the unshaded portion is the smaller population. So this smaller population is having the 9.5 kb 3' deletion, whereas the larger population is having mutation, large number of unique mutation described in LDLR gene that causes familial hypercholesteremia. So this is an example of founder effect, and founder effect is the loss of genetic variation that occurs when new population is established by very small number of individuals from larger population. So that is called as founder effect, and this is an example of founder effect. Then next one. In our body, skin cells and muscle cells share the same DNA. Which of the following is true? Although they share the same DNA, both have different genetic code. No. They don't have different genetic code, they have the same genetic code. The statement is incorrect. They are different tissues, so there is a difference in their DNA. They have given here they share the same DNA. So even they are different tissues, they share the same DNA. So statement B is also not correct. The statement is true. They differ though in the genes they transcribe and translate. Certain genes are transcribed in the skin cells and certain genes are transcribed in the muscle cell. It is not necessary that genes transcribed in the skin cells are transcribed in the muscle cell also. Similar and vice versa. So the statement C is more correct. Then next one. The statement is true. The skin cells and muscle cells differ in how many copies of DNA they have. Now you have always 23 pair of chromosomes. So that their muscle cell and skin cell have different copies of DNA is also not correct. Then next one. Which of the following techniques cannot be used to quantify affinity between an enzyme and peptide substrate? X-ray crystallography cannot be used for quantification. They have asked used to quantify affinity between enzyme and its peptide substrate. So X-ray crystallography cannot be used for the quantification purpose. Then next one. Which of the following statement is correct regarding the increase in molecular weight of proteins due to each of these post-translational modifications? So methylation, these are the post-translational modifications found in protein. So they have asked for increase in molecular weight of protein. So methylation will have less than acetylation. Acetylation will have more molecular weight than methylation. Phosphorylation will have more molecular weight than acetylation, and methylation will have more molecular weight than phosphorylation. So the answer here is A. The next one. You are carrying out an enzyme reaction in the lab and obtain Km and Vmax values. While repeating the experiment, you use only 1/10 of the enzyme. What would you observe? So here you have changed only the enzyme concentration. So what changes will you observe? You will see that Vmax would change with the change in enzyme concentration, but Km remains the same. Because Vmax depends on enzyme concentration, whereas Km doesn't depend on the enzyme concentration. So the answer here is option B. Then, rich fishing grounds, that is those that have high species diversity and population, such as those found in northern Japan or around Iceland, occur where cold and warm ocean currents mix. This is because upwelling of cold currents bringing nutrients to the surface, supporting phytoplankton blooms, providing food for many fish. That itself is the answer. Answer is option A. Then next one. Researchers passaged 3 different strains of E. coli. Black squares, they represent the wild type mutants. Red circles and blue triangles represent the mutants for several days. Gradually increasing concentration of the antibiotic cefatoxime N is equal to 96 replicate population per strain. Some population went extinct during the experiment. The plot below shows the number of surviving populations of each strain on y-axis as a function of antibiotic concentration on the x-axis. Panel A showed data for strains carrying one copy of beta-lactamase gene, and panel B showed data for strains carrying multiple copies of the gene. Given these results, which statement is incorrect? You have to find out the incorrect statement. Now, before coming to the explanation, let me tell you beta-lactamase enzyme, what it does is it inactivates cefatoxime antibiotic. This inactivation of the antibiotic provides resistance to the bacteria. Now, what have the researchers done? They have passaged 3 different strains of E. coli, that is wild type and mutant red and mutant blue. They have passaged. Passaged means they have like subculturing different strains of E. coli for several days, gradually increasing concentration of antibiotic cefatoxime. So they have used different concentration of cefatoxime, that is in increasing concentration range they have used, and they have subcultured this E. coli in increasing concentration of the antibiotic. So what will happen? Some E. coli strains will survive, some will die, like that. Now we will see the next question. Next, what they have told. Some population went extinct during the experiment naturally with higher concentration. You can see in this graph, that is panel A. This is the wild type. This is the survivors. So in this case, the survivors are decreasing and they have come to zero level. That is, they have gone extinct during the experiment. The plots below show the number of surviving population of each strain as a function of antibiotic concentration, that is x-axis. Panel A show data for strains carrying one copy of beta-lactamase gene. This is for one copy of beta-lactamase gene, and panel B is for multiple copies of the beta-lactamase gene. Now we will start reading from I will tell you the graph, what it means. So these are the wild type E. coli strains, these black ones. Over B and in A, these black ones represent the wild type E. coli. These are the survivors, what they have shown. So from 96 you have to start. So from there, with the increasing concentration of cefatoxim, you can see that there is a drastic reduction in the number of survivors, and it has come to almost zero in case of panel A. Whereas in panel B, it has not come to the level of zero. Okay, it has not come, it is somewhere between 0 and 16. So it has not become extinct in the case of panel B. Now why such a condition means? Because panel A is having only one copy of beta-lactamase gene, whereas panel B is having multiple copies of the gene. So it will produce more beta-lactamase protein. More beta-lactamase protein means it will start degrading the cefatoxim in the cefatoxim antibiotic, and the bacteria will become resistant to the antibiotic and it will survive. That is the difference. Then next thing I will tell you is these are the mutants. So here in panel A, mutants are also getting, they are somewhat nearing extinct condition. Mutant means one of the strains of E. coli that the researchers have used. Similarly in the case of blue also. But if you see panel B, the mutants have not gone into extinction very fast. Like as in A, when you compare it with A here, or even at higher concentration, that is even at 64 mg per liter concentration, the mutants are surviving in the cefatoxim antibiotic, both blue and red. This may be because either these mutants have multiple copies of the beta-lactamase gene, or it can be due to that certain mutations have arisen in these mutants that have led them to survive in the cefatoxim medium. Now read the options. 0.25 mg per liter cefatoxim is sufficient to kill half the wild type bacteria. If you see here, 0.25 mg is over here. Only 32 are survivors. That means somewhere around 64 have died. 96 minus 32. So somewhere around 64 have died. So how much are the survivors? Only 32, and 64 have died. So it is more than 50%. If they are telling that as per the question, 0.25 mg per liter cefatoxim is sufficient to kill half the bacteria. Half means 50% of the bacteria. But here it has killed more than 50% of the bacteria. Similarly, in case of B also, you can see that at 0.25, majority of them are survivors. Solely, that is more than 80 are survivors. So it has not killed half of the wild type bacterial population. So A is not a correct statement. Now next one. Adaptation to cefatoxim is enhanced by beta-lactamase gene. I already told you here, if you see both the graphs, Here it is reached almost extinction, but here the wild type has not reached up to extinction level. Similarly, the mutants are surviving. Have not reached up to the extinction level. They are surviving even at very high concentration of cephatoxin. So this can be enhanced. Adaptation to cephatoxin is enhanced by beta-lactamase gene, because B is having multiple copies and A is having only one copy. So this may be correct. So adaptation to CTX is enhanced by the beta-lactamase gene is correct. Copy number of beta-lactamase gene changes the probability of extinction. Of course, here they have gone in panel A, they have gone to extinction, whereas panel B, they have not gone to extinction very fast, even at higher concentration of or very high concentration of cephatoxin. So copy number changes the probability of extinction is correct. Then beta-lactamase is not the only factor affecting population survival. I told you there may be mutation in the mutants as well as in the wild type. That is why they have not gone under extinction as in panel A. And another thing that have enhanced the survival of these panel B strains is that they have multiple copies of the beta-lactamase gene. So beta-lactamase is not the only factor, but also mutations arising. Sudden mutations which are arising can also affect the population survival. So they have asked for the incorrect statement. So the answer here is A. Then next one. The mitochondrial F-type ATPase is a remarkable protein machine in which a subunit rotates during H+ transport and one ATP is generated for 120 degree of revolution. ATP hydrolysis releases approximately 50 kJ per mole and the transport of H+ cost about 19 kJ per mole. While the exact number depends on cell condition, approximately, this is your question. How many protons need to cross the membrane to achieve full revolution? Now, before coming into the details, all these numbers are not necessary. 19, 50 kilojoule, these are all not necessary. They have asked just to confuse you. So, before coming to the explanation, coming to the answer, I will like to explain certain things regarding F-type ATPase. Now this is the F-type ATPase. It is having this is F1 particle and this is the F0 particle. Now the F1 particle has alpha, how many? 1, 2 and 3 alpha subunits and 3 beta subunits, that is beta 1, beta 2, beta 3 beta subunits. And there is the gamma subunit which is acting as the central shaft and it will help in the rotation of this F1 particle. This is the F0. It is having A, then C, then B subunits. Okay. Now we will see the F1 particle. Now this is the section of the F1 particle. This one. This section. This arrowhead, it indicates the gamma subunit which is the central shaft and which will keep rotating. These are the beta, alpha, beta, alpha, beta, alpha subunits. Now this picture I have taken from Leninger, so explanation also has been very beautifully given in Leninger. You can go through it after hearing my explanation. Okay. So this is the model for how ATP is synthesized. Now first is that beta subunit, it exists in 3 conformations. They are designated as beta ATP, beta ADP and beta MT. Now what does this do? So in the first step, before this F1 subunit or F1 rotates, what will happen? So F1 is rotating because this gamma subunit is making contact with these alpha and beta subunits, and F1 is rotating, and due to this rotation ATP is released. Okay, now what will be the first step in this rotation? So first what will happen is ADP will bind to beta ADP. ADP plus PI will bind to beta ADP conformation. That will change the conformation of beta and it will get converted to beta ATP form, which can bind ATP, and again the beta ATP subunit changes to the beta MT conformation, which has low affinity for ATP, and it will be released to the enzyme surface. This is what is happening at 120-degree revolution. Now this is one 120-degree revolution. This is another 120-degree revolution, and this is another 120-degree revolution. They have shown it in 3 different pictures. But actually what is happening is once beta ADP is released from this beta ADP subunit, it will bind another beta ADP, then ATP beta ATP conformation will change and beta ATP will move to beta MT, and from beta MT ATP will be released. Now this region will be, this beta subunit will be having another bound ATP form, and this ATP again, once this ATP leaves the beta MT conformation, this ATP will enter in its place. That is how it is happening. That is what they have shown here. So this is for every 120 rotation, 1 ATP molecule is produced and 3 protons are needed. So this is the second 120-degree rotation, and another ATP molecule is released, 3 protons are needed. Again ATP is released, rotation of this F1 particle take place, again protons are released. So here they have told the mitochondrial F-type ATPase is a remarkable protein machine in which a subunit rotates during H plus transport and 1 ATP is generated for 120 degree of revolution. Now leave the next sentence. Then you read from here. While the exact number depends on cell condition, approximately how many protons need to cross the membrane to achieve full revolution? They have asked full revolution. Here only 1 ATP is generated for 120 degree revolution. So full revolution means 360 degree revolution. So for 360 degree revolution, how many ATP will be produced? 3 ATP molecules will be produced, and how many protons are needed? 3 protons here, 3 protons here, and 3 protons here. So totally 9 protons are needed. So the answer here is option D, that is 9. Coming to the next question. A peptide has the following sequence: A, T, H, M, A, R, A, M, E, N. They have given the alphabetic representation of amino acids. That is A, T, H, M, A, R, A, M, E, N. A is your alanine, T is your threonine, H is histidine, M is methionine, A is alanine, R is arginine, A is alanine, M is methionine, E is glutamic acid, and N is asparagine. So at pH 3.7, the net charge of the peptide is likely to be. So here you need not have to remember all the structures, but it is good if you can remember the structures. Especially the which all amino acid having side chain you have to remember. So when you are looking at the peptide, you have to take all the ionizable groups into account, both parent and side chain ionizable groups. Add up all the charges and you will get the net charge of the peptide. That is how you calculate the net charge. These are the parental ionizable groups. That is your alpha amino group and your alpha carboxylic group. These are the side chain groups which also could be ionized. So that also you have to take into consideration. Then next point is you have to remember the pKa values. I will not recommend that you remember all the pKa values of all the 20 amino acid. That is not possible. But I recommend that you memorize these. That is for histidine side chain pKa value is 6. For arginine, side chain pKa value is 12. For lysine, side chain pKa value is again 12. Then for the acidic amino acid, that is glutamic acid and aspartic acid, side chain pKa is 4. Then next you have to consider the pKa of the parental chain. So the pKa of alpha amino group is 10 and the pKa of the alpha carboxylic group is 2. If you remember these pK values, you can happily find out the net charge of the peptide. So first we have to find out if each ionizable side chain is protonated or deprotonated by comparing the pKa to the pH. If pKa is greater than pH, then it is protonated. The amino group will get protonated. If pH is greater than pKa, then it is deprotonated. Now we will see. First, we have to start from the alpha amino side chain. That is pKa is 10. Now at pH 3.7. So pKa is greater than pH. So it will be protonated. So there will be a great gain of H positive ion. So the net charge will be +1. Similarly, pKa 6, that is also greater than pH 3.7. So histidine also will gain a net positive charge. So the charge will be +1. pKa also for the arginine side chain is 12. So it will also gain a H positive ion. So the charge will be +1. Now coming to the carboxylic acid side chain. Here I told you for the glutamic acid, that is E, it is pKa 4 for the side chain carboxylic group. Side chain carboxylic group is having pKa 4. So this pKa 4 is somewhat approximately equal to pH of 3.7. So the net charge here will be 0. I have not put 0 over here, but it is taken as 0. I forgot to put it there over there. That is why. So pKa is 4. So the net charge of the side chain carboxylic group, this one is 0. Now coming to the side chain, to the alpha carboxylic acid, that is pKa of 2. That pKa is, so pKa, pH is greater than pKa. 3.7 is greater than 2. So it will be deprotonated. That is, it will lose the H positive ion to the solution. So it will have a charge of -1 because it is losing an H positive ion. So it will have a charge of -1. Now add all the charges, +1, +1, +1, 0, -1. You will get it as +2. So the answer here is answer option B, that is +2. Now one more thing I would like to tell you here is that these amino and carboxyl groups between the two amino acids, they are not taken into consideration because they are involved in peptide bond formation. So we need not have to worry about the amino and carboxyl groups between two amino acids. Now coming to the next question. A neuron takes 3 synapses on a muscle cell to produce a sudden movement. However, each synapse is unreliable and only conveys a signal with a probability of 0.5. Successful signal propagation by any of the 3 synapses will result in muscle contraction. What is the probability that the muscle will contract when the neuron fires? So, since each synapse is unreliable and conveys a signal with a probability of 0.5, the probability that a single synapse failing to convey the signal is 0.5. Okay, now you have to find the probability that the muscle will contract. So we need to consider the probability that at least one synapse succeeds in conveying the signal. So first you have to calculate the probability that all three synapses fail to convey the signal is 0.5 into 0.5 into 0.5, that is 1 by 8. Therefore, the probability that at least one synapse succeed in conveying the signal is 1 minus 1 by 8, that is 7 by 8. So when the neuron fires, the probability that the muscle will contract is 7 by 8. So that here is option A. In an in vitro transcription experiment, the transcript and both the strands of DNA were separated and analyzed for their base composition. The result obtained is shown in the following table. Determine which DNA strand is serving as template for mRNA synthesis. So here they have done an in vitro transcription experiment. Transcription is the formation of mRNA from DNA. The transcript, that is your mRNA, and both the strands of DNA, they were separated and analyzed for their base composition. So DNA strand 1, you got a base composition as given in the table for GATC. For DNA strand 2, you got a base composition as given in the table. U will not be there in DNA, it is present only in RNA, and RNA thymine will be absent. So this is your mRNA, thymine is absent here, and this is the base composition of various bases in mRNA. Now you have to determine which DNA strand is serving as template for mRNA synthesis. Now the template strand is called the non-coding strand. Now DNA, as you all know, it is having two strands. One strand and second strand. So this first strand is called as the template strand because it is the non-coding strand. The non-template strand is referred to as, this is the non-template strand, and this is referred to as the coding strand because its sequence will be same as the mRNA molecule. Okay. So mRNA molecule, that is if mRNA molecule is having A, then non-template strand also will have A at that position. If it is G at second position in mRNA, G at second position in the non-template strand. But here the question is for template strand. You have to see which is the template strand for mRNA synthesis. So if you see the base composition, you can see that DNA strand 1 and mRNA, they are having same base composition, that is G is 23.8 in DNA strand 1, 24 in mRNA, 30 for adenine, 29.6 for mRNA. T is absent in mRNA, but it is present in the form of U in mRNA. So 26 T in DNA strand 1 and it is almost equal to 25.8 U in mRNA. Then C is 20 and C is 20 in mRNA. So if you see DNA strand 1 and mRNA, you find that DNA strand 1, the base composition is same as that of the mRNA. So I already told you that the non-template strand is referred to as coding strand because its sequence will be same as that of the mRNA molecule. So here DNA strand 1 is same as that of the mRNA molecule. So DNA strand 1 is your non-template strand. So DNA strand 2 will naturally become the template strand. So the answer here is strand 2 because in question they have asked for the template for mRNA synthesis. So DNA strand 2 is the template for mRNA synthesis. And its base composition is not the same as that of the mRNA molecule. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you are new to my channel, please don't forget to subscribe it. I have also uploaded previous year TIFR solutions. You can go through my channel for the same. Thank you.